What are the mechanisms behind genome expansion or contraction? What role do genomic parasites, so-called transposons, play in this? And can the DNA content also change during an individual's lifetime? We will try to answer these and similar questions in today's episode of the series Secrets of Genes, in which I welcome you. My name is Edward Kajnachny, and I work as a geneticist at the Institute of Biophysics of the Czech Academy of Sciences. In one of the previous episodes, I talked about genome sizes. The size of genomes is determined by the interplay of various forces. Some of these forces lead to genome expansion, while others cause genomes to shrink. The increase in genome size, which we have already discussed, has apparently occurred especially over the last hundreds of millions of years, and it likely reflects the greater ability of larger genomes to respond to changing external conditions. A larger genome contains more basic modules that evolution can use to create and combine, mixing colors and their shades in the endlessly changing picture of life. The enlargement of genomes can occur in several ways. Perhaps the most elegant of these is the duplication of the entire genome, a process that scientists refer to as polyploidization. In the evolutionary lineage of some groups of organisms, this has even happened repeatedly. This phenomenon is especially common in plants. Most species of angiosperms have undergone at least one whole genome duplication in their evolution. And in the vertebrate lineage, genome duplication may have occurred even twice. And even a simple unicellular yeast bears traces of an ancient duplication of its genome. Whole genome duplication is a major shock in the evolution of a biological species. Afterwards, the entire genome becomes genetically destabilized, and many different rearrangements of genetic information occur. In some parts of the genome, sections are removed, as if the genome were shrinking back to its original size. In other parts, various rearrangements take place. And it is precisely in this rearrangement that the evolutionary potential of duplication lies. New genetic combinations can arise through these rearrangements. Evolution speeds up. Polyploidization is evolutionarily successful also because it increases the number of all genes equally, and thus does not significantly disrupt the balance between genes, which would happen if only certain parts of the genome, such as a particular chromosome, were duplicated. Another evolutionary force leading to genome enlargement is the duplication of certain regions within it. Sections of the genome that do not code for proteins can be duplicated, especially the already mentioned mobile genetic elements, or transposons. Transposons have sophisticated tools for self-replication, as we will see in the next episodes of our series. They are capable of occupying a significant portion of the genome in a very short time. Not only transposons, but also simple DNA motifs known as microsatellites, formed by repeating a few letters of the genetic alphabet, such as CA, 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 and so on, often expand in the genome. These often form long stretches within the genome. The genome also possesses mechanisms for removing some of its regions, thereby preventing an increase in its size. Transposons play an important role in these processes, and we know that they primarily increase genome size. So how is this possible? The trick is that transposons are present in the genome in a large number of copies. Transposons located in distant regions on the same chromosome can interact with each other due to their similarity in a process called ectopic recombination, which causes the region between them to be removed. It's comparable to the experience of having a very long rope with a vast multitude of various sticky spots, where far-reaching and distant sections can suddenly stick together, and then the entire region in between is eventually and completely removed. In this way, transposons act as elegant two-way regulators of genome size. Processes of genome reduction have been observed in the case of the Y sex chromosome. It is already known that over the course of evolution, it inevitably degenerates due to processes that we will explain in one of the upcoming episodes. This degeneration consists of gene erosion and the subsequent loss of larger or smaller segments of DNA. Genome reduction also accompanies the evolution of the genomes of cellular organelles, the so-called mitochondria and chloroplasts, whose genes are relocating from the organelles to the nucleus. I will also talk about these processes at some point. Parasite genomes are also subject to size reduction. A parasite simply gets rid of everything unnecessary in the interest of reproducing as quickly as possible. What it can take from its host, it no longer needs to encode in its own DNA, and it simply discards part of its genetic information. Changes in genome size can also occur during the lifetime of an individual. This can involve both an increase and a decrease in content of its DNA. A curious example is the silkworm. In its glands that produce silk, there are cells whose DNA multiplies up to 1,000,000 times. And all this is in an effort to produce as much product as possible, that is, silk. Here, it was advantageous to multiply the necessary genes by duplicating the entire genome. So that's all for today. In the next episode, we'll talk about what the genomic landscape of organisms looks like. We'll discuss how genes and repeats are distributed in the genome. I'm looking forward to seeing you. If you enjoy my storytelling, I'd appreciate a like. And if you'd like me to continue making episodes about genetics and evolution, please subscribe. This will help these videos reach more people. I really appreciate your support. Thank you, and bye-bye.